And so the final portion of the macromolecules, having already looked at carbohydrates, lipids, and proteins, are now the nucleic acids. Nucleic acids are macromolecules that store information, specifically genetic information. There are two different varieties of the nucleic acids. There's DNA, and as you can see, DNA is made up of a phosphate and sugar backbone. You can see them here. It goes sugar with the pentagonal shape, phosphate with the yellow ball, sugar, phosphate, sugar, phosphate. So this is the sugar phosphate backbone. And then there's also a nitrogenous base. There are 4 different nitrogenous bases that are going to be used in DNA: adenine, thymine, guanine, and cytosine. And these 4 are going to be found in numerous different iterations, and those are going to give us all of the individual variations that we have. So again, there are 2 types of nucleic acids. There's DNA, which is deoxyribonucleic acid, and there is RNA, which is ribonucleic acid. Both of them have a central role in the production of proteins. They're going to have a few differences, but this is once again showing the DNA with the adenine, thymine, guanine, and cytosine. And a nucleotide is going to contain the phosphate group, the sugar, and 1 base. So a nucleotide is 1 component of the nucleic acid. So the information that's stored in DNA is determined by the sequence of those bases. So in what order are the adenines, the guanines, the cytosines, and the thymines? So we see one particular sequence here, and if that varied, then we would have different information stored. Sort of like spelling out different letters in different orders will give us different words, and we can make different sentences and compose different books. So DNA is going to hold the genetic information that's going to be responsible for building an organism. It's going to make you human, and then specifically make you you. And one thing that is unique to the DNA, the deoxyribonucleic acid, is that it has this double helix structure. It's almost like a ladder that's been twisted up on itself. So you get the sugar phosphate backbone on both sides with the nitrogenous base sticking out of either end, and then there's a hydrogen bond that exists between the nitrogenous bases on either side of this ladder. And the base pairs are always going to bond in the same very predictable way. Adenine will always bond with thymine through that hydrogen bond. Guanine will always bond with cytosine. And what that means is, however many adenines there are within that DNA strand, there will be an equivalent number of thymines. Same thing with guanine and cytosine. The guanine and cytosine will be the same as each other, even though they might be different from the number of adenine and thymines present. So again, with these base pairs, they're always going to bond left side of the DNA rung to the right side of the DNA rung by a hydrogen bond that connects an adenine to a thymine, and a hydrogen bond that connects a guanine to a cytosine, or the reverse. So we'll have 2 complementary strands, 2 halves to the sides of the ladder that are going to connect through hydrogen bonds between these nitrogenous bases. So look at the strand of bases that you have here, and determine what would the complement be. What would be on the other side? If you know that A is going to always match up to T, and C and G are going to always match up, what will this other strand look like? And hopefully you realize that it should have been a G, 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 A, T, C, C, T, T, G, G. And we'll go over that in class. So RNA is frequently referred to as a translator. It's going to take the information that's stored in the DNA and bring it outside of the nucleus where the DNA is held so that proteins can be constructed outside of the cell's nucleus. And RNA is built in the very same manner to DNA, except instead of having two sides to this ladder, we're just going to have one side. It's like a half of a ladder, but just like the left half, not the top half. So we're going to have one sugar phosphate backbone working down. It's going to have the nitrogenous bases going across, but they're just not going to have the hydrogen bond to another half. Now it's going to happen that the sugar that's in there is ribose instead of deoxyribose, so we have a different sugar. There's only one part to this backbone, not two, and instead of thymine, we're going to have uracil. The other three nitrogenous bases are the same, but there is no T in the RNA, there's a U in its place. And this is sort of the fundamental rule in biology, that DNA is going to code for RNA, and RNA is going to code for proteins. And this is how pretty much all life on the planet functions. It is composed of DNA in some variety, which is going to have RNA that's going to translate that information and bring it to another part of the cell to make up proteins that are going to build that organism. So knowing now that you're going to start with DNA, we have two different strands here, hypothetical strands of DNA, and they're going to be turned into RNA. And remember, there's one difference with RNA. It's not going to have any T's. So your A's will not bond with T's. Adenine is going to bond with uracil instead. So we're going to have C's and G's, A's and U's when you make your RNA. So take a moment and look at these and find out what the RNA complement to this DNA would be. And here you go. This is what these sequences would look like. So I hope that you got these, and we'll be building these in lab.